Hello Year 11. This is lesson 4 in your booklet B2.4 Variation and Evolution. You will need your booklet in front of you and you need to turn to page 12. At the bottom of the page you should see a diagram similar to this, albeit in black and white. So this lesson is all about the different methods of cloning, and we're going to look at 4 different methods: tissue culture, taking cuttings, what we call adult cell cloning, and embryo transplants. So if we start with tissue culture, this is where if we take a plant that we find desirable and we want to create more of it, and we scrape a little bit of tissue from that plant, it can be anywhere, it can be a little section of leaf, it can be, as they're doing here, a section of the stalk, and we put it onto a sterile agar plate. So this little dish is called a Petri dish, and the Petri dish has some jelly growing in the bottom of it called agar, and the agar has some nutrients in that the plant will need to help it to grow. Particularly, it contains something called auxins. Auxins are hormones. Only the triple pupils will need to know this particular word, okay, because they've studied it previously. So we leave the tiny bits of scraped tissue sample from the plant on the agar for many days, and what happens is each of the pieces turns into a tiny plantlet. When we plant these into compost, they grow into the young plants, and then these are sold in the garden centres. So you will see as we're coming into the new year in the garden centres, when they start opening, that you will find these little trays of tiny little baby plants, and more than likely they've been created by tissue culture. So at the bottom of page 12, what I require you to do is to add these descriptions about what's happening, okay, into the boxes for me, please. So pause the video and add those in. Turn over the page now to page 13 and you should see this diagram. This is our second method of cloning plants, and it's called taking a cutting. So you might have parents, grandparents who do this at home, okay, to try and get new plants to grow. So say you've got a plant that you desire and you want to create multiple copies of it, okay? You would take a cutting of it. So you cut a piece off of the plant. Normally we cut with a bit of a slant, okay? And we would remove the lower leaves, okay? We want to minimize how much water is lost by evaporation and transpiration through these leaves are left. So you can see they've taken these leaves off the bottom and they've made sure that there's a good kind of like 1/4 of an inch, okay, of the stem and a slant. We then have to dip it into a special type of powder, and you can buy this in the garden centres. It's called root hormone powder, and what this does is it encourages the bottom of the stem to start setting out roots. However, this will take a few days to do so. So we then plant our cutting that's been dipped into the root hormone powder into some compost, some sterile compost here, around about 1 inch deep. And in order to prevent it losing water, because it hasn't got any roots, there's no root system at this point, okay, we cover the pot with a clear plastic bag. It has to be clear to allow light to pass into the bag and to the leaves so they can carry on photosynthesizing. And what happens is, because there are no roots to take up the water, any water that evaporates from the leaves, okay, will fall back down into the pot, okay, so that it keeps the soil moist until the root system has developed. Once we've started to get some tiny little roots here, then we can remove that plastic bag, okay, to allow the plant to continue to grow. What's important about this method is that this plant will be genetically identical to the plant it was taken from. That's the whole point of cloning. And so if we go back to our tissue culture, again, all of these plantlets will be clones genetically identical to the original parent from which they were taken from. So this is advantageous if we want to create a lot of exactly the same genetically identical plant. It makes production of plants cheap, okay, and easy to produce the same quality. So for example, tissue culture here is used to create something called oil palm. And there's an awful lot of oil palm that is used in lots of products, food products and in beauty products, okay? It's not necessarily a good thing because it's produced on a large scale as what we call monoculture. So often you find things like rainforests are cut down to grow oil palm because it can make more money for the farmers that are living in that particular area. It's used to grow bananas on a large scale. So if you've ever been out to the Canary Islands, to Tenerife, and you've driven around the islands, you may have seen huge big banana plantation. All of those banana trees would probably be clones in one particular area. And it's used as well, these are just 3 examples, there are many more, to create orchids. So orchids used to be considered quite expensive, exotic plants, okay, that only a few people could really afford. Now you can find orchids very, very cheaply in supermarkets, okay, and in florists, and particularly notoriously IKEA sells a lot of orchids, okay, that are cheap to produce and cheap to sell, okay. So there's some examples of our cloning of plants. For the page 13 then, all I require you to do is to put the right text, okay, underneath the diagrams. So if you haven't done so already, can you write these texts? Cut a new shoot from the parent plant. Remove the lower leaves off the shoot. That's number 1. Number 3, dip the shoot into root hormone powder. And number 4, cover the plastic, cover the cutting with a plastic bag until the roots grow. So pause the video, allow yourself time to write those statements underneath the correct diagrams for me, please. Now, we're now going to go on to look at adult cell cloning. I've already pre-recorded a short video from an animation that we use normally in school about the story of cloning Dolly the sheep. Dolly was the first adult mammal to be cloned, okay. Prior to that, we had cloned frogs, we'd cloned mice, okay, and even though mice are a mammal, they're quite a small mammal. So we wanted to know, could we clone a farm animal to be able to create, let's say, a whole flock of sheep that are all consistently the same, they're all genetically identical, they're clones, okay, so that we could create lots of meat very cheaply, okay, mass-produced in this way. This wasn't the only reason for cloning Dolly. If we were able to clone Dolly the sheep, then it meant we would be able to clone other organisms and animals. One of the main concerns that people had at the time was the cloning of humans, which is a definite no-no, okay? It is illegal in the UK to perform cloning experiments with human cells beyond so many weeks, okay? The science behind this was really, really important because it also gave us an insight into stem cells. And as we learned about with stem cells, the benefits of using stem cells for treating certain conditions and certain diseases, okay, is huge, okay? And potentially even creating organs that are bespoke to a person, an exact tissue match that can be used in transplants if we require them. So I'm going to tell you the story of Dolly the sheep again, even though you've seen a little video, and then you're going to use that information to help you create a task. So there were 3 sheep involved in creating Dolly the sheep. Normally in sheep reproduction is done sexually, so you need a male sheep, okay, to provide the sperm, and you need a female sheep to provide the egg. In the case of creating Dolly, all 3 sheep were female. And that's a really important point to begin with, okay? So they took a adult Finn Dorset sheep, okay, and they removed cells from their udders. The udders are the little teats down here that the baby lambs would feed off the milk from, okay? So they took cells from the udder cells of this sheep, a female, and we call these mammary cells. And they grew them up in a little petri dish, okay, and they selected a single cell here. The other sheep that they needed, they purposely chose a sheep that looked different. So they chose a different species called a Scottish blackface. This was going to be our egg donor sheep. So they removed an egg cell from the Scottish blackface. In order to do this, okay, they would have used IVF techniques, okay. So in IVF, in human IVF, they give the mother hormones to make the mother produce more than one egg cell. Okay, these hormones, okay, will act on the ovaries, making the ovaries mature more than one egg cell at a time. They can remove those egg cells then from our egg donor sheep here. And this is done using a needle and a suction, okay, a hollow needle and a suction to remove the egg cell. Once they'd removed the egg cell, underneath the microscope they could see the egg cell and the nucleus. And again, using a very, very fine needle, and this is techniques that they'd already learned about with IVF, they managed to remove the nucleus from the egg. So this egg is now empty. So we've now got an empty egg, which has been enucleated, okay? In other words, the nucleus has been removed. And we've got our single mammary cell that has DNA, okay, inside the cell itself. We combine the cells using an electric shock. So the DNA from the sheep's udder cell here, from the fin Dorset sheep, goes into the nucleus, okay, and combines. All of the DNA has come from this sheep. So there's no sperm and egg fertilization with half the DNA coming from the sperm and half from the egg. We now allow this cell to grow up, again in a petri dish, okay, into a ball of cells. So basically, this cell is going to be undergoing what we call mitosis. Remember, mitosis is for growth and repair. So this, in a way, is a little bit like a fertilized egg cell, with all the DNA coming from this sheep here. So the cells divide once to become 2 cells, twice to become 4, 8, 16, and so on until they become a ball of cells. And this ball of cells is called a blastocyst. The blastocyst has an outer ring of cells and an inner mass of cells here. So this will become a baby lamb, okay, in effect. This cannot grow outside the body. So this is where we need our third sheep. And our third sheep we call a surrogate, okay? A surrogate is a parent who's going to carry the embryo, okay, for however many weeks the gestation is for the animal. So humans can be surrogates. They can carry the embryos, okay, that have been created by IVF for a couple perhaps where the woman doesn't have a uterus. Perhaps she's had cancer and she's had her uterus removed, or perhaps her uterus has issues, is diseased, or has got a particular shape to it, which will mean the baby won't develop. So again, we use a surrogate ewe. Ewe is just the word we use for female sheep. Okay, ram is the male name. And again, notice that our ewe, they chose a sheep that was different to the sheep that they wanted to be cloned. So they chose the Scottish blackface to be the surrogate ewe. This was to make sure when the sheep was born, they could instantly tell, okay, if it was a clone of the fin Dorset, because it would have a white face as opposed to the black face here. And it would help them to understand what had happened. So the blastocyst develops into a fetus, and it takes about a 5-month gestation period for sheep to go through before the lamb is born. And Dolly was born, and she was an exact clone, okay, of this original sheep. So if you like, we've got sheep A, we've got sheep B, and we've got sheep C, and this is our cloned lamb that we have here. So using this information along with what you've watched in the video, so you might want to pause the videos, okay, this one and the other one, to help you answer the task or complete the task on page 14. So if you turn to page 14, you have got this diagram, okay? The sheep are just moved around slightly differently, okay? They don't start at the top of the page, okay? They're at the side of the page, but it is exactly the same process. I want you to add labels to your copy to explain what happens at each step to create the cloned sheep. I also want you to use the video that you watched on Cloning Dolly, the animation, to tell me how many sheep were used to create the cloned Dolly, which sheep is the lamb a clone of, and what colour face will the lamb have, okay? So make sure you use that important information, okay, in your diagram here. So pause the video and have a go at answering those questions, okay, and completing the boxes. So hopefully you've said from this sheep, okay, this is a sheep that's going to provide the donor egg. So remember what we said, this is removed using IVF techniques, and the donor egg is going to have the nucleus removed. If you can use the key terms that they've talked about, enucleated, then that's perfect. And I apologise for my dog woofing in the background, okay? But if you don't remember this word, don't worry, you can always just describe the nucleus is removed. In step 2 here's our white-faced sheep, okay? This is going to provide the udder cell that has been grown up, okay, in a petri dish. One of the things that they do with the udder cells is they turn them into stem cells. In other words, you'll hear them described in the video as totipotent. Please don't worry about using that word. That would be A-level and beyond, okay? But totipotent just means a stem cell, okay? In other words, a cell that can differentiate or specialise into any type of cell. So it's an unspecialised cell. They use an electric shock to join the empty egg cell with the udder cell here. So all the DNA is coming from our white-faced sheep here. Then this is allowed to grow by mitosis, dividing to create the embryo. And the embryo is inserted into the uterus of sheep C for 5-month gestation period. And the final product is that Dolly is a clone of sheep B. So she has a white face, okay? She's a clone. So make sure that you've filled in as much information as you possibly can on that diagram. Turn over the page to page 15. Now, I don't suggest you cut these statements out, okay, and the images out, because if you do, you're going to remove the work on the back of them. So the best way to approach this task is to number the statements on the left-hand column and the images that go with them on the right-hand column. Pause the video, okay, to give yourself a chance to number them 1 to 6, okay, and then I will go over the answers with you. So number 1, the DNA is extracted from the body cell of sheep A. Now you might be confused and think, well, this would be the diagram that will go with it. This is supposed to be a lamb, okay, compared with our sheep up there. So the picture that goes with it is this one here. Number 2, a nucleus is removed from the egg cell from sheep B. So it's this statement, okay, and the image is this one, because we can see that the DNA, okay, has been removed and we've got an empty egg cell. Number 3, the DNA from sheep A is fused with sheep B's egg cell. So that is this image here. So we're taking this DNA and we're putting it into that empty egg cell. It's a little bit easier on this PowerPoint because you can see the colour, okay, and the colour gives you a clue that this was the DNA from the egg, because it's a slightly different colour, because it would be a haploid nucleus. Number 4 should be the fused cell develops into an embryo, and that would be this diagram here. Number 5, the embryo is placed into the uterus of the surrogate sheep, sheep C, which that would be our picture here, the mum, okay, or the surrogate mum, I should say. And the lamb is born, it is a clone of sheep A, that's this diagram here. So make sure you've got those numbers in the correct place. If you've made a mistake, don't worry, just renumber them to check you've got the correct answer. There's then a practice GCSE question that I would like you to have a go of on page 16. So again, pause the video and have a go at answering this. There's a total of 5 marks. So if we go over this, this shows a method of cloning a sheep. We've got a black-faced female. In this case, they've used a white-faced male, and they've taken the body cell from the male this time. They've removed the nucleus from the egg, so they've enucleated it and fused the empty egg cell with the DNA from the nucleus of the white-faced male. This is allowed to divide into a cloned embryo, implanted into a surrogate mother, and is a cloned lamb. So for 2 marks here, it wants to know why the fusion of the body cell from the male sheep and the egg from the female sheep is an example of asexual reproduction. Remember, normally in sexual reproduction, we would have a sperm and an egg joining together. And in a human, there would be 23 chromosomes in both. And when they join together, it would create a cell that had 46, okay? So why is this asexual? Well, all the DNA has come from one sheep. The white-faced male. So only one parent's DNA is involved in creating the cloned lamb. You might say, well, there's also no sex cells involved. In other words, there's no sperm or egg, or you could use the word gametes. So there's 2 marks there. Give the gender is the sex, and the face colour of the cloned lamb. Well, if it's a clone of this one here, it's going to have a white face and it's going to be male. Give the reasons for your choice. Notice reasons, okay? Because all of the DNA, okay, will have come from the body cell from the male sheep. So all DNA is from body cell of male sheep with the white face. With white face. Okay, so there is no DNA from the female, from the black-faced female. And what they're looking for is that the offspring is genetically identical or a clone. Okay. Turn over the page then. We're going to look at the next method of cloning animals. So this diagram here, okay, is called cloning by embryo transplants. Normally at this point, okay, in the lesson, I would get out a piece of kit that was loaned to us many, many years ago from somebody who used to farm cattle, okay, and it is what we call an AI kit. Okay, unfortunately I'm able to show you that today, okay, but an AI kit, this stands for, okay, not artificial intelligence, okay, it's called artificial insemination. In other words, okay, taking sperm from one particular animal, okay, and putting it into the female, okay, of the same species of animal, okay. So sometimes with farmers, if they want to breed their cattle, it might be inconvenient for them to have to transport the female cow, okay, to another place in the country in order to be able to breed it with the male that they would like to be bred with. So there's actually companies that deal with selling frozen sperm from particular species of cattle, and then these are posted, okay, off to the farmer, and the farmer, along normally with a veterinary scientist, okay, veterinary doctor, okay, will help to inseminate perhaps the entire herd, okay, with some of the sperm that they have purchased, okay. So the reason we do this is we want to breed certain characteristics into our farm animals. We may want them, if they're dairy cattle, we may want them to produce a higher volume of milk, or a better quality of milk with more cream. If it's beef cattle, okay, it might be about creating a particular type of steak, a better quality steak, perhaps with either more fat or less fat, okay. So this is how AI is used, artificial insemination. So in order, okay, to create an entire field, okay, of cattle that are exactly the same, we would use not just AI, okay, artificial insemination, okay, but also IVF techniques too. So in this case, we would take the sperm from the male animal that we require. We would take the egg from the female animal that we want to breed with, okay? And so again, that would be done using IVF techniques. So the animal would have been given some hormones to encourage multiple egg production. The eggs would be removed. Each egg would be fertilized by a sperm. And that fertilized egg, that embryo, or zygote we call it to begin with, okay, will start to divide by mitosis to create an embryo. So all of this so far has happened outside of the body in Petri dishes like IVF. When we've got our embryo grown to a certain number of cells, what we can do is we can actually separate the cells into single cells, and then allow each separated cell itself to grow into an embryo in a separate Petri dish. Each of these embryos then is put into a surrogate cow. And so the reason why these cows all have like different markings on them, okay, is because the surrogates will all be genetically different. But what's important is that each surrogate mother will give birth to a calf, okay, that is genetically identical to the other calves, okay, from the other surrogates. It's not genetically identical to the surrogate mother. They're identical to themselves. So this ensures that you have the same quality, okay, of whether it's milk production or beef production from your entire herd. So you're creating a herd of cloned cattle, okay. So over the page, on page 17, you have got another similar kind of like matching task. You've got to, again, don't cut these out, okay, just number them. Match the statements in the left to the pictures on the right. This one is a little bit easier than the adult cell cloning one that we did previously. Pause the video and give yourself a chance, okay, to complete this task. So, the answers to this task. First of all, a prized cow would be given hormones to make it produce an egg cell. That should be produce, not product, okay? Produce an egg cell, and the egg cell is removed. So that matches with this diagram here. Number 2, the sperm from the prize bull, okay, which is often the prize bulls, their sperm is more expensive than others, okay? And it's used to artificially fertilize the egg cell outside the body to begin with. Question 3, the fertilized egg cell starts dividing to produce an embryo. So this would be this picture here. Number 4, statement 4, the embryo is split into 4. These start dividing into 4 embryos. So this would be this image here. Number 5, the 4 embryos are implanted into a surrogate mother, and the surrogates are all genetically different. And finally, number 6, that the 4 clone calves are born. So make sure that you've got those in the correct order, that you understand those. And our very last task to finish off with today, I thought this was on the PowerPoint, it's not. So page 18, you've got colour code the statements about embryo transplant in one colour and about tissue culture in another, and then number them to show the steps involved in each method. Pause the video at this point, give yourself a chance to have a go at answering that question, and I will go over the answers with you. So, if we colour code all the ones for tissue culture, we've got, first of all, a small piece of tissue is taken, okay, from the plant that you want to grow. It is placed in a substance to help it grow. That would be number 2 for tissue culture. Number 3, after about 12 weeks, roots and shoots start to appear for tissue culture. And number 4, the new organisms are planted for tissue culture. For embryo transplant, okay, first of all, we want sperm cells and egg cells are added to each other. That would be your number 1. Fertilization occurs for number 2. For number 3, each cell develops into an embryo, for embryo transplant. Number 4, the cells of the embryo. Sorry, I missed one out. Fertilization occurs. Number 3 should be the fertilized egg cell develops into an embryo. The cells of the embryo are split apart for number 4. Each cell develops into another new embryo. That would be number 5. And number 6, the embryos are placed into the wombs. So check that you've got those right. Apologies that I muddled you up, okay, halfway through. Okay, just pause the video, rewind and replay if you're confused over that one. Thank you, Year 11.